Actually, I was just going to take an in-depth look at it right now. Photosynthesis is the process in which plants make food. Plants capture approximately 5% of the sun's energy, and by photosynthesis make it available to almost every other living thing. The equation for photosynthesis is 6CO2 plus 6H2O plus light energy produces C6H12O6 plus 6O2. Photosynthetic organisms are largely self-sufficient. These organisms are also known as photoautotrophs. Autotrophs absorb carbon dioxide, water, and light energy from the surrounding environment, and by photosynthesis make sugar and oxygen.
Oh, thank you so much. Are we still lost, buddy? No, I think we're good. Thanks.
No problem.
Hello?
Hi.
Hi, we're a bit lost. Can you tell me anything about the Calvin cycle?
Actually, yeah, I was just studying some biology.
Great.
Come on over. Alright, so here's the Calvin cycle. Phase 1 is carbon fixation. 3 carbon dioxide molecules are attached to 3 ribulose 1,5-bisphosphate molecules with the help of rubisco, an enzyme. And then with the input of 3 H2O molecules, these become 6 3-phosphoglycerate molecules. And then 6 ATP becomes 6 ADP. Now they are 1,3-bisphosphoglycerate, and they have an extra phosphate. And then 6 NADPH becomes 6 NADP+, and 6 PI are released. Now we have 6 glyceraldehyde 3-phosphate molecules. This is phase 2, reduction reactions. Now 1 G3P molecule is released for each turn of the Calvin cycle. In phase 3, regeneration of RuBP, we have 5 G3P molecules left. With the input of 2 H2O, 2 PI are released. 3 ATP becomes 3 ADP, and now once again we have 3 ribulose 1,5-bisphosphate, and the cycle continues. For every 2 G3P molecules, we get 1 molecule of glucose.
Wow, this is awesome. I just have one question.
Yeah.
Where does this take place in the cell?
In the chloroplast of plant cells.
Oh.
And also, glucose stores the energy the chlorophyll originally captured from the sun.
Awesome. Well, you are a very knowledgeable stranger. Thank you for your help.
No problem.
I don't think we're lost anymore. So thanks again. See ya.
Great. Have a great day.
Oh, hey guys.
Oh, hey.
Aren't you guys the people who tell me all about photosynthesis in the park?
I think so.
Oh, this is awesome. Well, I was just doing some grocery shopping and
Us too.
wow, we should totally all go grocery shopping together.
Yeah, why not? Let's learn more about photosynthesis.
Yeah, we can teach each other about photosynthesis and everything.
Sounds good.
This is great. Let's go. Great idea. So I'm still curious about light reactions.
Oh, okay. Well, there's 2 types, cyclic and non-cyclic. So, cyclic uses only photosystem 1, whereas non-cyclic uses photosystem 1 and 2, and makes ATP and NADPH. Whereas cyclic just makes ATP.
Oh, I see.
So, there's 3 steps to the light reactions. 1, photoexcitation, the absorption of a photon by an electron of chlorophyll. 2, electron transport, transfer of the excited electron through a series of membrane-bound electron carriers, resulting in the pumping of a protein through the photosynthetic membrane, which creates an H+ reservoir and eventually reduces an electron acceptor. You see that?
Yeah.
Yeah, so, and then the next step is chemiosmosis, which is the movement of a proton through ATPase complexes to drive the photophosphorylation. And it's there where we get the ADP and PI to make ATP.
Great.
So overall, we have 3 ADP and 3 PI, which makes 3 ATP, 2 NADP+, and 2 H+, which makes 2 NADPH, and 2 H2O, which makes O2 and 4 H+. In non-cyclic. In cyclic, we just have ADP and PI, which make ATP.
That's it?
Yeah.
So in photosynthesis, there's light reactions.
Yeah.
So you're telling me all these plants that I'm holding went through light reactions?
Yeah.
That's awesome.
This is a table comparing the reactants and products of the dark reactions and light reactions. In the Calvin cycle, we can see that 9 ATP, 6 NADPH, 3 CO2, 5 H2O, and 3 RUBP are the reactants. The products are 9 ADP, 6 NADP+, 1 G3P, 8 PI, and 3 RUBP. In the non-cyclic light reactions, the reactants are 3-ADP and 3-PI, 2-NADP+, and 2H+, and 2H2O. The products are 3-ATP, 2-NADPH, O2, and 4H+. Now, in the cyclic light reactions, the reactants are only 1-ADP and 1-PI, and the product is 1-ATP. 4H+ are pumped into the stroma, which helps with the concentration gradient and makes ATP synthase turn, therefore creating the ATP. As we can see, no NADPH is formed in the cyclic light reactions.
Wow, Bo, I learned so much today. I learned all about photosynthesis and light reactions and everything. And I did some research just now, and I even found out ways of alternative mechanisms of carbon fixation. I know, interesting, isn't it? So basically, Rubisco is an enzyme that may catalyze two reactions: the addition of CO2 to RuBP, or the addition of oxygen to RuBP. When there's more oxygen than carbon dioxide, oxygen binds more often to RuBP, resulting in the oxidation of RuBP, instead of its carboxylation to PGA. I don't know if you knew this, but photorespiration occurs in the light. And photorespiration decreases the production of carbohydrates by photosynthesis. This means that alternative mechanisms must occur in order for the plant to undergo carbon fixation. The first alternative we're going to look at is C4 plants, or C4 photosynthesis. Thousands of plants undergo this photosynthesis. Well, an enzyme called phosphoenol pyruvate carboxylase, or PEP carboxylase, catalyzes the addition of carbon dioxide to a 3-carbon molecule called PEP. This makes a 4-carbon molecule called oxaloacetate, and since that's a 4-carbon molecule, that's why it's called C4 photosynthesis. The leaves contain two types of photosynthetic cells, the bundle sheath cell and the mesophyll cell. The mesophyll cells are located around the bundle sheath cells. The PEP enzyme fixes carbon dioxide by catalyzing the attachment of CO2 to 3-carbon PEP, forming 4-carbon oxaloacetate. Oxaloacetate is converted to malate, and malate diffuses from the mesophyll cells into bundle sheath cells through cell-to-cell connections called plasmodesmata. Carbon dioxide is then removed from malate, which is decarboxylation. This whole reaction is endergonic and uses 1 ATP. The carbon dioxide that was removed from malate in the bundle sheath cells enters the C3 Calvin cycle in a second fixation reaction. This time, it's catalyzed by Rubisco in the bundle sheath cell. The second alternative to carbon fixation is CAM. Plants such as cacti undergo CAM because they are in a hot and dry and sunny environment. These plants have their stomata open at night, and then they convert carbon dioxide into glucose during the day. So that's pretty much it for carbon fixation.
Well, that was an eventful day.
Yeah, it sure was, but I didn't get to talk about the chloroplast.
Oh, well, go ahead. I'd love to hear.
Great. Well, the chloroplast is an organelle in plant and algal cells that carries out photosynthesis.
Cool.
They contain chlorophyll, which gives leaves and stems their characteristic green color. Well, the chloroplast has two limiting membranes, the outer and the inner membrane. The membranes are enclosed in an interior space filled with a protein-rich semi-liquid material called stroma. A system of membrane-bound sacs called thylakoids are stacked one on top of the other to form characteristic columns called grana.
Cool.
Photosynthesis occurs partly within the stroma and partly within the thylakoids membranes. Chloroplasts, like mitochondria, contain their own DNA and ribosomes. This allows them to replicate by fission.
Cool.
If the concentration of CO2 goes up, the rate of photosynthesis goes up as well. But light intensity limits the products from light reactions. Dark reactions cannot go faster. That is why the graph levels off. If the concentration of CO2 in the air goes up, the CO2 uptake goes up as well. This is also limited by the light intensity. As oxygen production increases and as light intensity increases, the rate of photosynthesis increases as well. Temperature is a limit to the amount of oxygen released during photosynthesis. The ideal temperature for photosynthesis is at 25 degrees Celsius.
Well, that was a great day. I love when we get to talk about photosynthesis.
Me too. If only we had a cool way to remember all that we've learned.